Assalamualaikum and greetings to all examiners. My name is Shariffah Fadhilah binti s52264 and my final year project was on isolation and characterization of fungi from disease ornamental fishes in Terengganu area. And my supervisor is Dr. Nurul Akila Ibrahim. For background of study, Malaysia was enlisted the fifth largest producer export value at 22.62 million in 2014 by day 2016. And the major group of farm-breed ornamental fish are usually goldfish, barb, tetra, swordtail, and also platyfish. And for climbing perch, it also famous because of its survival at low dissolved oxygen. For during import, export, transfer, and transit, the fish are usually expected to be injured, and extreme fluctuation in water quality temperature can lead to fungal infection by 2016. And most notable are Sapoletnia, Archaea, Amorphosite species. And Aspergillus also found infected in certain organs, both in external and internal. For problem statement, fungal disease are the second most serious cause of losses in aquaculture by May 1921, and fungal known to attack to all life stage of fish from egg to adult. For the significance of study, poor water quality can also lead to an increased fungal infection and with wide range of infection depending on the management of the farm itself. For the objective of study is to isolate fungi from disease ornamental fishes and to identify using microscopic and microscopic in fishes in Kuala Terengganu area. For the method and material, for site selection, the sample were obtained from local market at Pasar Tani Padang Giliran, Kuala Terengganu, Malaysia. And the sample species was selected were climbing perch and platyfish. For fungi sample collection, the fish body were divided into two parts, that is exterior and interior. For the culture media preparation, we have two. That is, the first one is PDA preparation, where the PDA agar powder was mixed with distilled water and autoclave for 2 hours, and then wait for it to solidify in the laminar flow machine. And for the antibiotic agar, is the PDA agar mixture mixed with antibiotic, that is chlorophenicol, to avoid unwanted bacteria to grow on the media. For the isolation of fungi, the fungi were divided into two phases, that is culture media and isolation to pure culture media. And the plate will inoculate at 28 to 30 degrees Celsius and note at the 7 days and to observe the different colony and unique colonies. For the method and material objective 2, microscopic, the microscopic is to identify the morphology of characteristic for colony, color of the colony, and the surface area. For the microscopic, the morphology study involves observing spore, hyphae, coniospore under a compound microscope. For the fungi preservation, the slime were made by using PDA agar mixture mixed with antibiotic, and the slime were placed at 28 to 30 degrees Celsius for it to grow. The result were expected genus Mucor, Penicillium rhizophus, and 2 samples were Aspergillus and 1 sample expected bacteria colony. For sample A, for microscopic identification, the colony was cottony white, reverse colony was yellow pigmentation. For microscopic, we can see there are spongiform and sporangium. For sample B, the colony were glabrous and white. And for microscopic, they are bacillus and streptobacilli shape. For sample C, the colony was green with white ring, and the reverse were brown. For microscopic, they are conidospore and hyphae. In sample D, we can see that the colony was white, cottony, and yellow pigmentation in reverse. And for microscopic, they are columella with apophysis and sporangium. In sample E, the powder green texture, cottony back pigmentation on top. And in microscopic, we can see there are long conidophore and conidoradial chain. In sample F, white wrinkle heap on top with pale yellow reverse colony pigmentation for macromorphology. And for microscopic, we can see there are conidospore with vesicle and conidospore. The sample expected genus was A. Mucor, B. Bacterial colony, C. Penicillium, D. Rhizophus, E and F was Aspergillus. And those genus were referred article by this article, that is Mucor, Penicillium, rhizophus, and aspergillus. For discussion, this study was carried out morphological method to identify fungi isolate from disease ornamental fishes. And the isolated species belong to two classes, that is Rhizomycete and Zecomycete. Aspergillosis animal were also infected in the fish, and also poor water quality enhanced growth of fungi in water body. Thus, a good husbandry in field can help. And infections such as Panacea, Mucor, Aspergillus, microbial contamination does not usually give a result in disease, but also an environmental stressor and give a high potential pathogen to their host. To, as, to conclude, as this study is very likely that Aspergillus genus will present in climbing perch fish sample, and there are large variation and distribution of fungus species in different locations and also different source of adaptation. And for proper management and control of these fungi can reduce fungal infection. This study recommended that further work be done such as PCR procedure and histopathological tests in future to isolate and identify more accurate or filamentous fungi for taxonomic pathogenicity investigation. This is my reference. Thank you for lending me your ear.